So in this video, we shall discuss about glomerulus. Kidneys have a special type of tubes called nephron, and these tubes are involved in the formation of urine. In two kidneys, we have nearly 2 to 2.4 millions of nephron. Every nephron has a glomerulus, which is the filtering unit. Our main discussion will be about the structure and function of the glomerulus. So what is a glomerulus? Glomerulus is mainly the filtering unit for a nephron. As you know, we have nearly 2.4 millions of nephron in both the kidneys. It means we should have nearly 2.4 millions of glomeruli in both the kidneys together. But here I will concentrate on the concept of one nephron and one glomerulus first. Okay. So first of all, we'll see the structure of glomerulus first, and then we will see how glomerulus functions as the filtration unit and what are the factors which alters the filtration through the glomerulus structure. Okay. So let us suppose the glomerulus structure. Suppose here I draw an afferent arteriole which is bringing the blood to the filtration unit, that is the glomerulus. Then you know what will happen. This afferent arteriole, it will break down into multiple capillaries. Then these capillaries rejoin one another to make the efferent arteriole. Okay, so what we really see, there is afferent arteriole which is bringing the blood, and afferent arteriole break down into bunch of capillaries, and these anastomosing channels of capillaries are called glomerular capillary. Okay, so this is a glomerular capillary tuft. I have shown only 3 loops of capillaries here, but in actual it breaks down into many loops of capillaries, and these anastomosing channels meets up to make efferent arteriole. Okay, and these glomerular capillary tuft, it is very specialized capillaries, because you can see they are fed by an arteriole and even they are drained by an arteriole. So they have a very high pressure system. Next, the glomerular capillary tuft, it is surrounded by or invested by a double layered cells. Okay, so these are a very specialized double layer cell. This is the first layer known as the visceral epithelial cell, and this is the another layer. Okay, and this is the parietal epithelial cell. Okay, so we have got visceral epithelial cell and the parietal epithelial cell, that means the double layer of epithelial cell below the glomerulus. And here we have got, yes, this is our basement membrane. Okay, and then these parietal epithelial cell, they join together to form the next group of cell, that is the proximal convoluted tubule, that we'll study later on. Okay, so this double layer of epithelial cell, it makes a capsule known as the Bowman's capsule. Okay, now another important point which I want to mention is that there is a specialized type of cell present in between these capillaries. They are known as the mesangial cells. Okay, so this mesangial cells, they are present in between the capillaries, and these mesangial cells, they are producing a connective tissue. Okay, so these mesangial cells produces a lots of connective tissue, and this connective tissue along with the mesangial cell together, they are known as mesangium. Okay, now you should know what is a glomerulus. Glomerulus is the group of anastomosing capillaries present in the nephron, invested by double layer of epithelial cells that makes the Bowman's capsule, and the capillaries are embedded within the mesangium, and whole the glomerulus is functionally the filtration unit of the nephron. Okay. Next, we'll see the whole structure into detail one by one. Now, let's suppose here it is a glomerular basement membrane, and on one side of the glomerular basement membrane will be the blood flowing membrane, that is the capillary side. And to make it simple, the blood is coming from the afferent arteriole and the blood is draining into the efferent arteriole. Here we can represent cells over here. Okay, so these are the endothelial cells and these are the mesangial cells along with the mesangial connective tissue. Okay, then on the another side of the membrane we have got cell, that is the visceral epithelial cells and the parietal epithelial cells that makes the Bowman's capsule, and next everything is drained into the proximal convoluted tubule. Okay. Now first we shall study in detail about the endothelial cell. Okay, the blood side, the endothelial cells. The endothelial cells that lines the glomerular epithelium, it is very specialized type of cell. And what is very special thing about it, the endothelial cell which is present over here, they have got thousands and thousands of holes within the cell. Okay, so we have got so many holes within the endothelial cells, and such endothelial cells that have got holes and perforations within the cells, such type of cells are known as the fenestrations, and such group of capillaries are known as fenestrated capillaries. Okay, the holes are known as fenestrae or fenestrations, and such type of capillaries that have got fenestrations, they are known as fenestrated capillaries. Now, what is the size of these holes or these fenestrations? So these fenestrations which are present over these endothelial cells, the size is approximately 70 to 100 nanometer. Now, because this whole layer has to act as a filtration system, we should know that filtered material has to pass through these fenestrations. Okay, therefore, glomerular endothelial cells has thousands of perforations or fenestrations. Each are about 70 to 100 nanometer, and these perforations enhances the filtration process. Okay. So this was the special thing about the endothelial cells. Okay, perforations, fenestration. The endothelial cells have got holes. This is the important thing about the endothelial cells. Next, we will talk about the glomerular basement membrane. When we look at the glomerular basement membrane under an electron microscope, you will find that the central area is very dense. This dark central area of the glomerular basement membrane is known as the lamina densa. Okay, and on both sides of the lamina densa, the area of the glomerular membrane looks less dense. We say that the central area is strongly electron dense, but on the endothelial side and epithelial side of the basement membrane, it is relatively electron lucent. Electron lucent means under electron microscope the area looks less dense. Okay, and it is called lamina rara. Okay, the lamina rara towards the endothelial side is called lamina rara interna, and the one towards the epithelial side is known as lamina rara externa. Okay, now let's go into further detail about this structure, that how exactly the glomerular basement membrane is made. Actually, the glomerular basement membrane structure is primarily made up of collagen type 4. Okay, so this collagen that makes the suprastructure and basic network of glomerular basement membrane, this collagen molecule, it consists of triple helix of alpha chain, and this triple helix peptide chain is making the collagen type 4. Okay, and this millions of collagen type 4 is making the basic structure of glomerular basement membrane. Okay, I hope you people are getting. This collagen type 4 is made up of three, you can say three lines over here, pink, green and blue. So they are the triple helix of alpha peptide chain. So collagen type 4 is made up of triple helix alpha peptide chain, and this collagen type 4, lots and lots of collagen type 4 put together, it forms the glomerular basement membrane. Okay, and this part of the peptide chain, this is known as the globular non-collagenous domain of the peptide. Okay, then another important thing, now you know that throughout this structure, that is the lamina densa, type 4 collagen is present within the glomerular basement membrane. Okay, now another thing that I will tell you, which is very important, is that this type 4 collagen is making the suprastructure within the glomerular basement membrane. Okay, and over this there is deposition of many negatively charged molecules, and these molecules are polyanionic proteoglycans. Moreover, there are laminins, fibronectins, antectins, and you need not to remember all this. Just remember that the glomerular basement membrane is electronegative. At the top, even there are special negatively charged molecules present not only in the glomerular basement membrane, but negatively charged molecules are also present on the endothelial cells, as well as they are also present on the epithelial cells. And these negatively charged molecules as a group, they are known as sialoglycoprotein. Okay, so what we really see is there is electronegativity within the basement membrane. There is also electronegativity present on the endothelial cells and electronegativity present on the epithelial cells. That means the whole filtration unit is electronegative. Okay, so this was all about regarding the glomerular basement membrane. The important thing about the glomerular basement membrane is they are made up of collagen type 4, and the second thing is they is very much electronegative. Next, we will talk about the epithelial cells, that to the visceral epithelial cells. Okay, so it is very important to know that these epithelial cells, when they are healthy, they have got food processes. Okay, and with the tip of these food processes, these cells are attached with the glomerular basement membrane, and these processes not only help to hold with the basement membrane, these processes even interdigitate with one another and with the next cells. Okay, like suppose these fingers are like digits. Okay, this is a visceral epithelial cells and the fingers are the digit. So the food process of one cell will be interdigitating with the food process of the another cell. Okay, so these processes, they interdigitate one cell to the next cell and forming a longitudinal slits known as the filtration slit. Okay, so now you should be able to know it in a much clearer manner. Okay, so this thing, as you can see, this is the filtration slits. Okay, these are the filtration slits. Okay, and on the endothelial side, what we had? Yes, on the endothelial side, we had fenestration, isn't it? Here we have fenestration on the endothelial side, and on the epithelial side, we have got filtration slits. On the endothelial side, the size of these fenestration was nearly 70 to 100 nanometer, isn't it? But the size of the filtration slit, it is 20 to 30 nanometer. It means the size barrier. Okay, fenestrations are larger and slits are narrower. Therefore, molecules that are smaller can pass through it, and molecules that are larger, they cannot pass through it. Another point to remember is that these visceral epithelial cells only have got the food processes. Okay, food processes, these digits, these are present only and only in the visceral epithelial cells. Food processes are not present in the parietal epithelial cells. Next, a substance Suppose this is any substance flowing in the blood, it has to pass through first through these fenestrations. Okay, first of all it passes through the fenestration, then they crosses the electronegative basement membrane collagen type 4, and then they move laterally and then through the filtration slit. Okay, now very recently there is a concept that these filtration slits, as you can see over here, these filtration slits, they have got a thing known as filtration diaphragm. Okay, over here and over here also we have got filtration diaphragm, and this filtration diaphragm is made up of a special protein known as nephrin. Okay, now what we have studied, anything that has to cross from the endothelial side, okay, that has to cross from the endothelial side to the Bowman's capsule, it has to be big in size or small in size? Yes, small in size, isn't it? Larger molecules cannot move. We have seen when you keep on increasing the molecular weight of a substance, the chances of that substance of getting filtered are getting less. So we have got a classical example of dextran. Dextran molecule, it can be synthesized of different sizes, small size dextran and big size dextran we can synthesize. Even we can synthesize dextran molecule of different charges, okay, with positive, neutral and negative charge. So green means positively charged dextran, blue means neutral dextran, and red means negatively charged dextran. Then they did the experiment that when we infuse intravenously dextran molecule, very small size dextran molecule, it freely filtered, and as they increase the size, filtration will become less. Due to this reason, they say that this glomerular filtration barrier is a size barrier, because filtration depends on the size of the molecule to be filtered. Smaller molecules like glucose and amino acid, they freely filter, but plasma protein, which are larger molecules, they cannot filter. Okay. Secondly, they did another experiment. Okay, that first they injected dextran molecule that could freely filter. Okay, then they made these molecules electropositive. Okay, the filtration will increase or decrease. What do you think? See, if the molecule have right size to be filtered and at the top they are positively charged, that means they are cationic molecules. So what do you think? These cationic molecules will be repelled from the membrane or facilitated? Yes, they will be facilitated because the membrane is highly negatively charged. Okay, the basement membrane is highly negatively charged. The endothelial side is highly negatively charged. Even the epithelial side is negatively charged. And so any molecule that comes here and that molecule is positively charged, it will be attracted and moved through it. Okay. So if suppose we have got, no, if there is a group of dextran molecule and one group is positively charged and another group is negatively charged, okay, but this group of dextran molecule, they have got same molecular weight, all of them have same size, but just one is positively charged and another one is negatively charged, then among this, best filtration will be seen for cationic dextran and worst filtration will be seen for anionic dextran. So it means that filtration not only depends upon size but also on the charge. Smaller size molecule will filter more and larger size molecule will filter less. Cationic molecule will filter more and anionic molecule will filter less. That means anything that has to pass through here finds two types of barrier. One is the size barrier and another one is the charge barrier. That is why we say that glomerular capillaries acts as size and charge barrier system for the filtration. Now, let me give you a very classical example that you have to remember. You know, in the blood we have got RBCs, WBCs and platelets. isn't it? RBCs are very large, around 7 to 8 micron. Next, WBCs are around 12 to 14 micron, and platelets are around 1 to 2 micron. And all these are so large that in a healthy glomerular capillary system, none of them can filter. It means that when the blood is passing through this filtration unit in a healthy system, RBC, WBC and platelets, they cannot filter due to their very large size. They are prevented from filtration due to this size barrier. Next, many of the things present in the plasma, they freely filter, but in a healthy glomerular membrane system, filtration system, plasma protein do not filter. You know why? First of all, what are plasma proteins? Plasma proteins are like albumin, globulin, and fibrinogen. Albumins are the smallest protein, globulin are larger, and fibrinogens are largest. Actually, they have seen that the size of this albumin molecule is just right to be filtered, but still it could not filter. What could be the reason for that? Because albumin molecule know that their size are just exact to be getting filtered through this size barrier, but the problem is albumins are negatively charged molecules, okay, and for this reason, even the size is small, then also the filtration is being inhibited. Okay, next we'll have few words about the mesangial cells. They are active players. They have special properties, like first of all, these mesangial cells, they are contractile cells. They can contract. So if they can contract and they are present around the capillaries, it means they have some influence on the capillary filtration. Next, the mesangial cells can phagocytose. They act as phagocytic cells, so that whenever there is antigen antibody complexes here, these cells can engulf them. And lastly, mesangial cells can proliferate also. Okay, so this was a little information regarding mesangial cell, and this was all about our today's discussion regarding the glomerulus. In my next video, I will be talking about the glomerular filtration rate. I hope you people found this video helpful. Thank you for watching.